And let me introduce next speaker, Sam Sharifi. He's the head of research at Matter Bioworks, the company which is doing DNA protection research. Sorry, I cannot do two things at once, apparently. Yeah, Sam is a great researcher and a good friend. So he has been in the industry for more than a decade as well. PhD in aging biology in Leipzig, so also a good veteran of the industry. Please welcome Sam.
Okay, thanks for the intro. So at Matter, we want to intervene at as upstream as possible. So we are focusing on the DNA. So what we've all learned is that you have one genome and every cell in your body has the same genome. But we know in reality, that's true in theory, but in reality it's every cell is unique and has a different genome. So we have trillion genomes, and this is because being alive causes damage. So DNA is actually a very unstable molecule. We know this from the lab in the tube, it's very unstable, but in the cells it's actually not much different. So there are like internal processes and external processes that damages your DNA continuously. And your body is actually pretty good at repairing this, but over time you will accumulate damage, and your DNA repair mechanism also goes down with time. In addition, if you want to think of long-term space travel, space is also very damaging for your DNA. So if you want to do something about it, we need to protect our DNA before going to space. You don't want to arrive on Mars with an increased risk of cancer. So normally the DNA repair mechanism is very good, but it still makes mistakes. And if it makes mistakes, you incorporate a different sequence, and this we call mutations. So we either have point mutations, a single change in the code, or larger structural variants. And one example was also telomere attrition is, for example, also a structural variant. And what's interesting is recently there was a study of Alex Kagan showing that there is a correlation between lifespan and your mutation rate per year. So if you look at a mouse that lives very short, it accumulates per year a lot of mutations, whereas a naked mole rat that lives much longer has a very slow rate. And this is very well correlated. So, and in addition to that, it seems that all of the organisms are within a range of between 2,000 and 5,000 mutations at the end of life, which means that there is kind of a cap of amount of mutations you can have. So you accumulate mutations, but not all of these mutations are bad. So some of them are neutral, some of them don't do anything. You have, of course, bad ones, like the worst ones we know are tumors from cancer. Some just go into apoptosis, and then you have a lot of other cells that kind of are not directly pathological, but over time they might affect functions of clonal cells. So what we want to do is like bring your genome back to baseline, so kind of remove all of the genomes that are not your original germline and bring it back. So that's the end goal. And of course this cannot be done with one pill, so we have a different assets to do this. So first we want to understand what's going on, so we want to read the genome. So we have, and the technology for this was not available until a couple of years ago. With normal NGS that's been around for a long time, you cannot see the differences between cells. And now we have a new form of sequencing, which is called error-corrected sequencing, where we can distinguish the differences between cells. And so we have this technology called single molecule mutation sequencing. It's called SMM-seq, where we can highly accurately detect on each molecule the mutations that are there. And here also, this is in humans, so the somatic mutations have been correlated with age. And recently there have also been published a clock that's kind of similar to the epigenetic clock. So then we have a genetic clock where you can look at the somatic mutations and see what is your age. So the next would be to remove cells, also overly corrupted cells like tumors. It's not good to try to reverse them or to treat them. It's just better to kill them off. So for this, we developed a listeria, an attenuated listeria that can specifically go to tumors. So and it can carry any payload. And so we deliver to the tumor, it affects the tumor and then delivers the payload. And for our own internal program, we deliver tetanus antigens. So we will make your tumor look like tetanus, and then your body knows what to do because you're vaccinated against it, and it will remove it. So the indication that we wanted to go first for was pancreatic cancer, and pancreatic is one of the worst cancers because it's detected so late and it's very deadly, and there's no cure for it. So what we did then in mice, in stage 4, like metastatic pancreatic cancer, we kind of treated them with the listeria, and what we then see here is like a very nice reduction of the tumor size, but even better, that the metastasis was also decreased. And here you see a kind of an image where normally you get this swelling, and which then is disappeared after treating it. And of course, the next question is, does it also help these mice live longer? So we did a survival curve of this, and we see a 40% increase in survival upon treatment. Now, this method should be tumor agnostic, so all tumors have this microenvironment where the listeria can go to. So we thought, okay, let's test another indication. So we tested then stage 4 ovarian cancer. And here we also see that you get a 54% increase in survival with our listeria treatment. Now, these mice, they have been genetically engineered that they all the time produce tumors, so they will be never cured. So basically, they die because of the tumor again. So we thought, okay, what if we treat them again? Will that extend their survival? And then upon treating them again, this is you cannot see it very well, but this is the line here. It basically doubles their survival. So it means that we could keep on treating you if you get tumors every time. So the treatment is still effective. Now, so this will go now into clinical trials now. So for next year, we'll start a phase 1/2a clinical trial in pancreatic cancer patients. So after you've removed all of the corrupted cells, so the next thing you want to do is kind of look at the other cells, and they have mutations, and we want to then replace these parts of the genome. So for this, we want to develop an editor, and what we are using is a transposase. And what is transposase? So it's an enzyme that helps transposons. So transposons are jumping genes, kind of they move around in your genome to kind of insert in different sites. So we kind of use a transposase, but transposase is not targeted, so we kind of put a Cas on it to make it targeted. So briefly to go over the mechanism here. So you would have the transposase with the Cas, so you have the editor here, binds to the donor DNA, then the Cas finds the guide, and the guide guides it to the target site, where it then inserts the larger insertion. And the advantage of using transposases is because there is no size limit in principle. Just to show you some preliminary data, so what we did did a test with puromycin. So puromycin is toxic to the cells. It causes premature chain termination of the nascent translated protein. And what we did is we want to insert puromycin resistance genes that kind of detoxifies puromycin, and then check if it improves the survival of the cells. So normally the cells would die. So you kind of if you're not treated, and if you already have if you don't suffer from puromycin resistance, you would survive. So we treated them with our editor at different concentrations, and then treated them with puromycin, and we see this improve in survival of the cells in different variants that we created. And of course, the work is not done there, so we now will have to optimize it. This is all a little bit small. So what we want to do is to optimize because we want to reduce the amount of off-target, especially if you want things used for longevity, and improve the on-target. So we can do this via two ways. One is it's called random mutagenesis, so it's directed evolution. We cause mutations and let nature do the work for us, and then put a selection pressure on it to then screen for it. The other way would be just to look at the structure and kind of do it rationally and then look computationally what is the 3D structure and where should we make the mutations to improve its function. So the last thing would be, of course, or the ideal case would be to not even get damage, right, and to enhance repair. So to do this, we looked at people that are already good at this, those are the centenarians. Oh, before I show the centenarians, I want to show you this. So there was already a very old study that showed that if you increase P53, P16, and third in the genome, that kind of already gets your a very remarkable lifespan extension in mice. And this is a very old study already. So we can, using gene therapy, extend lifespan there. So we want to start safe, and centenarians are safe because those are genes that are safe in humans and already used. And we are mainly interested in genome stability and DNA repair, and we see that the majority of the 300 known genes that have been involved in the longevity of centenarians, that their majority are that they're in processes that are involved in genome stability and DNA repair. So, of course, we talked also about talked a bit about the business model, and of course, like just giving people centenarian genes is not a viable business model. So what we want to do is look at these genes that are in centenarians and also look for a disease indication where these genes are important in. And you have some. And interestingly, some of these diseases are also accelerated aging diseases, so as Werner syndrome, and you have progeria, and so this would be a kind of our first indications. So this is how the current pipeline looks like. So the sequencing assay is already commercially available. We have CRO partnerships, pharma clients. The immuno-oncology therapy goes into clinical trial next year, and the editor and centenarian program is currently in R&D phase. So this is our team of experts, so the scientific co-founders, Claudia Gravenkamp being a stereo expert, Jan and Alex are very well known in the somatic mutation field, and George, of course, being an expert in gene editing. And we have also two very good experts in Yanan and Deso, Chris Solis from eGenesis and Rejuvenate Bio, who are helping us with gene editing. Okay, that's it. Thank you. Any questions?
Cool. When and where do I get my DNA repair transposon shot?
Honduras. No. Maybe in a couple of years.
In a couple of. Would you do that?
I wouldn't see the off-target being very low. Yeah, then I would do it. Yeah.
Okay. Do you do like any animal trials with this already?
No. This is still in cell lines first. Yeah.
Can you talk more about corporate strategy with cancer? Because kind of like we are on longevity conference, and a lot of longevity companies suddenly doing something for cancer. What's your company strategy with these cancer assets, and what's with the longevity assets?
Yeah, I mean, we're happy to partner with the cancer assets as well, because I think you anyway cannot bring it you can only bring it to a certain stage yourself, right? So, but the longevity asset we really want to develop ourselves, and then even within the longevity assets, like for example with the editor, we would happy to be partner out for certain clinical indications, but we want to have it for longevity for ourselves. Yeah.
Good. Linus?
Thank you for a great talk. Super impressive technology. Would you speak some more about the general business model? Like, how are you gonna make money on this?
Yeah, that's a good question. I mean, with biotech, it's in general like you develop it to a certain stage and then you sell it off, right? And so the advantages with gene editors is kind of now it's so hot and there are so many indications that you can go off that you can partner in principle or sell just for certain indications of, and that would be a good way to make money, and then just keep developing what you want, the indication you want to do yourself. But it's also, do you want to become a big pharma and do phase 3, phase 4 yourself, right? Because a small biotech doesn't have the infrastructure to do that. So you have to think about kind of, do I want to develop the infrastructure to start doing really big clinical trials or?
Yeah.
So basically you do a single indication, age-related disease, up to like phase 1, maybe phase 2, and then you sell it off to big pharma basically, and that's how you make the money.
Yeah, but you just have to be careful that you don't get distracted too much, because that happens with a lot of the longevity companies. They start very like ambitious as a longevity company, and then down the road they get investors that have certain they have priorities, and then they just become a standard biotech company. Yeah. And I think your pick of investors is also important there. So you really need people that believe in your vision, because they don't push you out of this road. No, you have to make money for me, right? Like so. Yeah.
Hey there. Thanks so much for the extensive presentation of your work and your teams as well. Really appreciate it. I have a two-part question. First is, when you say human trials, do you actually mean simply in vitro with human cells from pancreas, or do you actually mean inoculation?
It means actually in humans. Human trials means that these are terminally ill pancreatic cancer patients that the treatment, we do it as a second-line treatment, so that the first treatment didn't work. They don't basically, there is actually no good treatment for pancreatic cancer, so then...
Okay, good. No, I'm asking because I come from the field of neuroscience, and sometimes when we have human trials, when we're talking about dementia Alzheimer's, we just retrieve some cells and then we do it in vitro. But thanks for clarifying.